Taxonomy for Hornbill, Kingdom Animalia, Phylum Chordata, Class Avis, Order Bucerotiformes, Family Bucerotidae, Genus Buceros, Species Buceros rhinoceros.
For physical characteristics, this bird large, approximately 80 to 90 cm in length. It has distinctive banana-shaped cast on top its beak and used for attracting mates. Male also have larger cast than female for the habitat found in tropical rainforests of Southeast Asia, especially Malaysia, Indonesia, and Thailand. These birds also prefer dense, undisturbed lowland forests. For their diet, eat primarily frugivorous, feeding on fruit, especially figs, insects, and small animals. Why this species is chosen? First, ecology are important. As a keystone species, the rhinoceros hornbill play a crucial role in seed depressor, promoting forest regeneration and maintaining biodiversity in tropical rainforests. Second, threatened status. Classified as vulnerable on IUCN Red List, this species face significant threat from habitat loss, hunting, and illegal pet trade, making its conservation urgent. Third, conservation potential. With target conservation effort and genetic management strategies, there is a strong potential for successful recovery for rhinoceros hornbill population. Lastly, unique breeding behavior. The hornbill nesting behavior, where females seal themselves inside the tree cavity, make them particularly susceptible to habitat destruction.
The genetic conservation plan for the rhinoceros hornbill focuses on in situ in the wild and ex situ outside of natural habitat strategies to preserve and enhance genetic diversity while addressing threats to the species. The first is protect and expand existing lowland rainforests in Malaysia, such as those in Borneo and Peninsular Malaysia. Create wildlife corridors to reconnect fragmented forests, enabling hornbill populations to maintain genetic diversity. Next is regularly conduct genetic studies to assess the health and diversity of populations. For example, analyze DNA from feeders or droppings to monitor genetic structure. Utilize sound-based monitoring systems to estimate population densities by recording and analyzing their calls. Next, the last is work with local and indigenous communities to raise awareness of the hornbill's ecological importance and reduce poaching and habitat destruction. Promote sustainable ecotourism centered around birdwatching, benefiting local communities and creating incentives to protect hornbills and their habitats. For ex situ conservation, first is establish breeding programs in reputable zoos and avian sanctuaries to ensure genetically diverse and healthy populations. Utilize artificial incubation to support egg survival, especially in situations where natural nests are threatened. For cryopreserved genetic materials such as sperm, eggs, and somatic cells, this will ensure that the genetic material of diverse populations is available for future breeding or research. Store samples from diverse populations to capture genetic variability. The last is reintroduce captive bred hornbills into areas where populations have declined significantly, ensuring habitats are suitable and protected. Use genetic profiling to ensure new individuals contribute to genetic diversity.
In situ technology for conservation hornbill. Number 1 is artificial net box, designed to mimic the temperature and humidity of natural tree cavities, attracting hornbill to nest to allow the hornbill to live inside. Number 2 is GPS tracking, mounted on hornbill to monitor their movement, breeding territories, and habitat use. These data are important for understanding their ecological needs and informing management strategies. Number 3 is reforestation project. Plant native tree species that provide food and nesting site for hornbill. This alternative focus on the redemption of forest fragments that help to restore habitat that are important for the survival of hornbill. Ex situ technology for conservation hornbill. Number 1 is capture breeding program. Establish breeding program in zoo and wildlife sanctuary to maintain genetic diversity. This program can use advanced veterinary care and nutritional management to ensure a healthy population. Number 2 is gene bank. cryopreservation technique can be used to store genetic material such as sperm or eggs for hornbill. This genetic bank can serve as a resource for future breeding program or reintroduction effort of this animal. Number 3 is public engagement platform. Using social media and mobile application to engage the public in conservation effort can increase awareness and support for hornbill conservation initiative.
Possible factors causing failure of the program and mitigation measure. First, habitat degradation. Continued deforestation and habitat fragmentation could undermine conservation efforts. To mitigate this, strong legal framework must be enforced, coupled with community-led initiatives that promote sustainable practice. Second, genetic bottlenecking. In captive breeding program, a limited number of individuals may lead to reduced genetic diversity. To prevent this, genetic monitoring should be conducted regularly. Ensure that breeding pairs are selected based on genetic compatibility. Third, human-wildlife conflicts. Encroachment on hornbill habitat may lead to conflict with local communities. Addressing this requires implementing compensation schemes for crop damage caused by hornbill and promoting coexistence strategies. Lastly, insufficient funding. Conservation programs often face budget constraints. To counteract this, partnership with NGOs, government agencies, and private sector should be fostered to secure funding for ongoing conservation initiatives. Lastly, for conclusion, the rhinoceros hornbill is a vital species facing significant threat on habitat loss and hunting. A comprehensive genetic conservation plan that integrates both in situ and ex situ strategy is essential for ensuring its survival. By leveraging modern technologies and fostering community involvement, we can enhance conservation efforts aimed at protecting this iconic species while maintaining biodiversity in Southeast Asia, the tropical forests.